Good afternoon, everyone. My name is Barbara McMakin, and I'm from the NINDS Office of Neuroscience, Communications, and Engagement. On behalf of the NIH, I would like to welcome you to this afternoon's call and to thank you for your interest in participating in this discussion with us today. Today's call is being recorded. If you have any objections, please disconnect at this time. Unfortunately, Dr. Koroshetz has a conflict today and can't join us for this call. After some updates from NIH, Dr. Vicki Whittemore will introduce the guest speakers who will give a presentation on their recent work. Then we will open the webinar to questions. During the Q&A session, if you have a question for our speakers, please select the raise hand button at the bottom of your Zoom screen and we will call on you to unmute. If you are joining us by phone, please dial *9 to raise or lower your hand and *6 to mute and unmute. You can also submit questions using the Q&A box at the bottom of your screen. We will try to make our remarks brief so that we can answer as many questions as possible in the time we have available to us this afternoon. Now, I would like to hand the call over to Dr. Vicki Whittemore.
Thank you, Barbara. Good afternoon and welcome to the second NIH ME CFS call for 2023. Since our last call, there have been several developments in NIH-related activities regarding ME CFS, and we look forward to sharing all of those updates with you. I'll start with updates on the activities of the Trans NIH ME CFS Working Group, and then turn it over to Dr. Nath, who will give updates on the Intramural ME CFS studies and other studies ongoing in Intramural NIH. Then Dr. Joe Breen from NIAID will update us on the Collaborative Research Centers and Data Management Coordinating Center, excuse me, and talk about the upcoming NIH conference that is the planning's underway for. And then we'll have two guest speakers who I will introduce. So we're pleased to have Drs. Gunnar Gottschalk and Avik Roy, who I'm pleased to announce was just awarded an NIH R21 Research Grant Award last week. So congrats to both of them and thanks for being here. So first, I'd like to update you on the process we're going through to develop an NIH ME CFS research roadmap. So the goal of this roadmap is really to assess the state of the research and to determine what we know, what we don't know, and identify gaps in the research that will really help to drive the research toward clinical trials. So it's, as everyone knows and is well aware, there are no FDA-approved treatments for ME CFS. And so what we would like to really focus the research priorities that come out of this research roadmap on are what is needed to really get to clinical trials that we can develop new treatments and FDA-approved treatments for ME CFS. So we have invited and appointed individuals to serve on the NINDS Working Group of Council. So this group is made up of several clinicians, investigators, as well as leaders of the ME CFS advocacy community, as well as 5 individuals with lived experience. So they're either individuals who themselves have ME CFS or are caregivers for individuals or an individual or individuals with ME CFS. In addition, we, through a self-nomination process where we were quite overwhelmed with a large number of applications, which was really fantastic to have so many people interested in participating, we identified 27 additional individuals with lived experience. And again, these are caregivers or individuals with ME CFS to also participate in the process. And they'll be helping us to organize and carry out several different webinars that will be open to the public on different topics in areas of ME CFS research. And those webinars will be open to the public. And we're, like I said, just in the initial planning phases of the first webinar that we hope to have scheduled sometime in June. So we certainly will make the community aware of the date and information about that webinar as soon as it's available. So there will be ways for all of you who aren't actually part of this process already to participate. So as I said, through the webinars, we're also going to use a crowdsourcing tool called IdeaScale to gain additional input from the community on the research priorities, because after each of these webinars, the group will develop research priorities from the information that's gathered listening to subject matter experts at the webinar to put out research priorities for that area. And so we'll be looking for additional feedback from the community. And then a draft report will come out at the end of 2023 to be presented to NINDS Council in February of 2024. I also want to share that we've awarded a grant for the NIH Center for Collaborative Research at Cornell University and are close to issuing the award for the Data Management Coordinating Center. We also reissued the funding opportunity, and it's our intention to fund an additional 2 collaborative research centers. And the deadline for those applications is mid-June, June 16th. We're also in the process of organizing a young investigator meeting that will take place the day before the NIH Research Conference, which Dr. Breen will talk about later. The Young Investigator Workshop and the conference will be both in person and virtual. And so we're still working out the details for both of those. But again, we'll let you know as soon as the details are available for those meetings. With that, I'd like to turn it over to Dr. Nath.
Thank you, Vicki. So I'm pleased to share where we stand on our studies with regards to ME/CFS. So as you know, we conducted a very comprehensive large study here intramurally, and it's was not an easy task to put it all together and to even write it because there were so many different disciplines involved. We've now put it together and the manuscript has been submitted. It's currently under review. So that will have explicit details on everything that we did to this population. However, everything cannot go into a single manuscript. So there'll be several other manuscripts that are going to come out. Each one of them is going to take a piece of it and then describe it in more detail. So a second manuscript on muscle alone has already gone out, is under review. And then there'll be one on microbiome and so on and so forth. So there's going to be a whole host of manuscripts coming out as soon as the first one gets accepted for publication. We're also keen on trying to see, based on these observations, if we should try to conduct some pilot clinical studies, clinical trials here at NIH. So those things are currently in process of discussion. And we've done a lot of work on long COVID over this last 3 years. And we've submitted several manuscripts that have already been published, one describing the peripheral neuropathies, one transverse myelitis, 2 major manuscripts on neuropathology of COVID, one on cognitive aspects of long COVID that is currently in press. I just saw the proofs yesterday. And we've written lots of reviews, editorials, shared the information with the public and scientific organizations. We've organized workshops on the topic that includes ME/CFS and long COVID. So we've tried to get the community engaged the best that we can. We also initiated a study on Gulf War syndrome, and those patients have started coming in. And lastly, we also have a clinical trial for long COVID using IVIG, and so we're currently recruiting patients for that study. So I'll stop here. Thank you.
Thanks. Over to you, Dr. Breen.
Thanks, Dr. Whittemore. Hi, my name is Joe Breen. I'm a program official in NIAID and work closely with Vicki and others in NINDS on the NIH-wide ME/CFS working group. And I want to report a little bit of the progress that's occurred in the collaborative research centers since we last spoke at the last telebriefing. There really are 2 major areas that I want to talk about. One is papers that came out actually just about at the same time as the last telebriefing, and this was 2 two studies that were done by the Columbia group led by Ian Lipkin, and also the Jackson Lab group led by Julia Unematz and Julia O, that really ended up characterizing the gut microbiome from ME/CF patients and healthy controls, and showed that there were significant differences in butyrate producing capacity. In both papers, in fact, found the same, as well as different bacterial species that differed. But what was nice is that it was validating, and that it happened in two centers. Both were reported independently in Cell Host and Microbe. But I think it lends more evidence to the metabolic disturbances, as well as helps to highlight some of the pathways that could potentially be intervened in a clinical study, for example. And I think something that was lost in some of it was the phenotypic and microbial and even metabolic biomarkers that come out of these studies, which in a research setting, I think will help direct future work. Coincident with these reports, actually, the center led by Dr. Maureen Hansen at Cornell had two papers that I want to talk about. One was published very recently in March by Jeff Moore, is the first author, looking at really post-exertional malaise in ME/CFS subjects and healthy controls. And this was a study in actually a significant number of patients, 80 patients and 64 healthy controls, showed that after a 2-day exercise testing, that those with ME/CFS took about 2 weeks to recover, whereas the healthy controls took 2 days. Now, this is interesting for a few reasons. It may help us use a quantitative measurements of PEM, which could actually end up helping design strategies to manage, but also knowing this kind of information can be really helpful to inform people who go into future studies in terms of what outcomes that can be expected. So I think we can't broadly overinterpret it, but I think it was a really unique finding. And the other unique finding from the Hansen group is a study that's a small pilot, but showing that there were differences in urine metabolism between female ME/CF patients after maximal exertion versus healthy controls. So I think that's also very interesting. It's obviously a readily accessible fluid for biomarker studies, et cetera. Something that isn't a pilot scale, but will definitely, and I'm sure is being followed up. And the Collaborative Research Network, as Vicki mentioned, has a data coordinating center, and they published a paper in 2021 showing their MAP ME/CFS tool. And I would encourage folks to return to that website, because they are really working hard and adding new data and really increasing the power of that tool as part of the data management and coordinating center function. I think I'll stop there. I think that's the general progress for the CRCs. And I just wanted to follow up that the NIH working group is planning on a meeting in 2023. It will be in December 12th and 13th on campus. Really, as a follow-up to the 2019 meeting, this will be a way to talk about the status of ME/CFS research, but also link it to potential interventions based on pathways which have been discovered since 2019 and subsequent, and also leverage to the extent possible and learning we can from long COVID. There's a organizing committee that's creating an agenda and co-chairs, and we're very thankful to have co-chairs, Drs. Nancy Klimas and Andrew Grimson. So well, as soon as the agenda is public, of course, we'll share that. And it will, again, like Vicki mentioned, it will be a public meeting and also hybrid. So while we have it on campus, it should be available wider as well. Over.
Great. Thank you very much, Joe. So I see a couple of people have hands up. We're going to take questions after we hear from our guest speakers. So not that we're ignoring you. We see that your hands are up, but I'll start the question and answer period after we hear from Avik and Gunnar. So it's my pleasure today to introduce Dr. Avik Roy, who's the Chief Scientific Officer at Simmaron Research Incorporated, and Dr. Gunnar Gottschalk, who's the Executive Director of Research at Simmaron Research. And as I said earlier, we're thrilled that NINDS has just issued a research grant award to Gunnar and Avik entitled ATG 13, a new player in ME/CFS. And so we're very pleased to have you both today, and I'll turn it over to you for your presentation. So thank you both.
Hello, everyone. Thank you so much for the invitation from NIH. My name is Dr. Gunnar Gottschalk. It's so wonderful to be here to share some of our recent work and to introduce what our plans are moving forward in the next couple of years. Simmaron Research is a 501c3 not-profit biomedical research institute. We have and really try to focus on multidisciplinary translational science. We collaborate directly with Dr. Dan Peterson at Sierra Internal Medicine, where for many years we've worked and collected samples and executed clinical trials in Incline Village, Nevada. Our more recent change has been to bring and start the Simmaron Research and Development Laboratory, which is based at the University of Wisconsin-Milwaukee campus. And our primary focus there is to translate some of the findings from the Incline Village and other clinical collaborations into the laboratory. And that includes looking at biomarkers and animal models, as well as drug discovery. A huge benefit of our spot at University of Wisconsin-Milwaukee is we are also a part of the Milwaukee Institute for Drug Design. So that is a group that essentially is a group of chemists that is focused on drug development in multiple different types of disease settings. And so while we're very much new to the UWM system, and certainly chronic fatigue syndrome, ME/CFS is very new to the UWM system, we're extremely excited to be there and leverage opportunities to try to find new mouse models, new cures, etc. This is our team. You'll be hearing from Dr. Avik Roy, who's the Chief Scientific Officer following this. Of course, Dr. Daniel Peterson is still the clinical PI at Sierra Internal Medicine. And I am myself, as well as the Executive Director, a Principal Investigator. We have two primary focuses coming up in 2023 that Avik will get into in a moment, looking at mTOR activation and autophagy impairment. We've published a manuscript looking at the target ATG 13, which is a very important molecule for the induction of autophagy. Autophagy is a process in the cell that helps remove specific types of proteins from intracellular waste products. And that is one of our projects. And then secondarily, we're trying to translate that finding of autophagy impairment in ME/CFS patients into animal model development, specifically looking at cardinal symptoms of ME/CFS. I want to preface this by saying we're not trying to make a mouse model to understand all of ME/CFS. That would be impossible. Our primary focus is to look at cardinal symptoms, including post-exertional malaise. We feel that in other fields of neuroscience, including Alzheimer's disease and Parkinson's disease, there has been a lot of progress made with respect to drug development and the understanding of illnesses through animal models. And that's what we plan on doing. And specifically because both Avik and myself have a background in Parkinson's disease and Alzheimer's disease, we felt that it would be a wonderful contribution to this field. Like Vicki said, we're very thankful for the R21 funding. We've also had support from the Solve ME Ramsey Award program grant, which is another competitive grant award system. And also through our local connections to the Parasol Tahoe Community Foundation. And finally, a thank you to all of the donors who also support our work. Thank you so much. And with that, I'm going to turn it over to Dr. Avik Roy, and he's going to talk about our current progress. Avik.
Dr. Roy, you're muted.
On behalf of Simmaron Research, I would like to say thank you to NIH for this opportunity. Today, I'm going to talk about our ongoing research. It's in very pilot stage right now. How mTOR activation and subsequent autophagy impairment are critical in the context of ME/CFS. So let's start with some background. ME/CFS is a chronic multi-system illness characterized by severe muscle fatigue, pain, and brain fog. Cardinal symptoms of this disease are post-exertional malaise, which is a severe fatigue, pain, and dizziness post-treadmill exercise, and orthostatic intolerance, which is characterized by sudden drop of blood pressure while maintaining the upright posture. Despite intense investigation, until now, the molecular mechanism of this disease is poorly understood. However, there are some potential contributing pathways that are believed to be involved in the pathogenesis of ME/CFS. Some of these are mitochondrial toxicity, upregulation of inflammatory molecules, myelin abnormalities, and autophagy impairment. There are challenges that impede the understanding of this disease are lack of sufficient postmortem biospecimens. This disease is highly heterogeneous in nature in terms of its etiology, and there is no reliable disease model. So our goal is to make a disease model to study some of the pathological features of ME/CFS. In our context, it is post-exertional malaise. So we want to achieve this goal by 3 different strategies. The first one is a drug-induced animal model in which we will administer drug in a mouse to induce PEM. The second is transgenic mouse model, where we manipulate a host gene to induce post-exertional malaise. And number 3 is a disease-relevant model where we want to induce a virus or a plasma-infused mouse model. So let's talk about a drug-induced mouse model first, because this is the study we are doing right now. So the targeted pathway is autophagy. So let's give a brief background of autophagy. Autophagy is a cellular quality control process by which the defective mitochondria and biologically inactive proteins are enclosed in a vesicle known as autophagosome and then targeted to lysosome for further hydrolysis. So if you look at the entire process, you can see that there are multiple proteins that are involved in the process in different layers. And many of these proteins are tightly connected in disease and regular health. For an example, like Beclin and LC32, these 2 proteins are reported to be upregulated in the serum of patients with acute ischemic stroke. Beclin and P62, these 2 proteins are upregulated in the serum of patients with pulmonary embolism. ATG5 has been reported to be upregulated in AD patients, Alzheimer patients. Alpha-synuclein is reported to be upregulated in Parkinsonian patients. So many of these proteins are upregulated in serum during different metabolic disorders. However, the role of this pathway, autophagy, and its impairment is not studied well in ME/CFS. So to understand the autophagy impairment, in ME/CFS patients, we performed an antibody array. Antibody array is a procedure in which a membrane is engraved with antibodies of different metabolic proteins. So in this, the membrane is engraved with 34 antibodies of 34 different autophagy-related proteins. And after that, that membrane is challenged with the serum of healthy control and patients. So we have two different representative images. These are collected from two different case control studies. The first one is like age-matched and gender-matched healthy male and patient. And the second one is age and gender-matched female patients and healthy control. So in both cases, you can see that the myriads of proteins, there are a lot of autophagy-related proteins that are upregulated in patient serum. However, the ATG13, which is shown in this rectangular enclosure, in both cases, consistently upregulated. So we are intrigued about the upregulation of ATG13 in patient serum, in ME/CFS patients. So subsequently, we performed an ELIJA study in 10 different cases, like 10 healthy controls and 10 age-matched patients. And we saw that ATG13 is consistently upregulated in the serum of patients, in the serum of all like ME/CFS patients. We further continued to understand the metabolic regulation of ATG13, and we did immunoblot studies where we found that ATG13 is heavily phosphorylated in its serine residue, and that phosphoserum is strongly elevated in patients in ME/CFS serum. So we further continue to show that the ATG13, when it is phosphorylated, it's become inactive, and it goes to the serum of ME/CFS patients. And it also can induce a ROS and nitric oxide production via RAGE-mediated induction of INOS. We published this piece of work in Molecular and Cellular Neuroscience last year. But our current study is just after that, like it starts just after that. So until now, we know that ATG13 is inactivated, and when it gets inactivated, it becomes phosphorylated, and it is elevated in the serum, and it causes autophagy impairment, and that subsequently causes the other downstream metabolic disorders, such as mitochondrial dysfunction, cell death, and inflammation. But we really don't know what causes ATG13 inactivation. So what is the upstream molecule that causes ATG13 inactivation? So then we look at the signaling pathway, and we found that there is a cytosolic protein. Its name is mammalian target of rapamycin, or mTOR. It's a kinase. It's a serine threonine kinase, and it has a dual role in the cell. So when it gets inactive, then ATG13 can participate with the autophagy, and that is very integral process for a healthy tissue. But when mTOR is active, then it phosphorylates ATG13 in multiple serine residues that causes the inactivation of ATG13, and ATG13 eventually aborts that active complex formation, and that causes autophagy impairment. And we also hypothesize that mTOR, since it's a kinase, it can regulate many downstream pathways that can induce a different inflammatory molecules in the muscle tissue. For an example, IL-6 and RANTES, and that might contribute to the pathology as well. And we want to test this hypothesis by this compound, and we put the code name that SIM501. The SIM501 is a potent mTOR activator, and we want to see that what happens if this compound is administered in mouse. So we took 3 different doses, like 2, 5, and 10 milligram per kilo body weight. And we administered this drug in mouse via gavage, via oral route, for 14 days. And after 14 days, we plan to measure muscle fatigue through EMG measurement, post-exertional malaise, pre- and post-treadmill regime, And we also want to see the attention deficit and brain fog in these mouse. Interestingly, after 5 consecutive days of feeding, we saw there is severe weakness in young female mice. So these female mice, they show severe fatigue with periodic episodes of freezing. Their movement is slow, and at the end of 14 days of feeding, they show a severe loss of their movement, severe fatigue, and a hypothermia. So we are going to the next slides where we can discuss more about this. So after 14 days of feeding, what we found that the young female mice, they didn't gain body weight, and they have sufficient loss of muscle mass. And when we performed EMG analysis, we saw that the SIM501 treated mice, they display a certain pattern of muscle wave, which we call the marching soldier muscle wave. And that kind of muscle wave we see when there is inflammatory demyelination in muscle tissue. So muscle tissue are getting innervated by different peripheral nerves. So when there is demyelination in these nerves, then we see this kind of muscle wave. Whereas for the vehicle-fed mice, we see there is like a normal, a regular kind of muscle wave here. And we also measured grip strength. And what we saw that increasing doses of SIM501 caused a severe loss in grip strength. And we also measure overall movement in the mice, and we saw that yes, that SIM-treated mice, they have shown a significant loss of their movement with periodic freezing. Now we wanted to see the molecular evidences, like what exactly is happening in the tissue level. So we performed H&E staining just to see that mononuclear infiltration in the tissue. And we found that in biceps muscle in those mice, there is severe infiltration of mononuclear cells. And when we performed, when we characterized that what kind of T cells or what kind of mononuclear cells are infiltrated, we didn't see neither CD4 nor CD8 T cells were there in the muscle parenchyma. However, we observed that there is a severe infiltration of macrophages in the muscle tissue after SIM treatment. We further characterized those macrophages because there are 2 different types of macrophages. One type of macrophage is inflammatory and toxic. We call it M1 macrophages. And the other macrophages, they're called M2 macrophages, which are kind of protective, and they're anti-inflammatory. And what we found that those infiltrated macrophages, they have expressed CD40, which is a marker of M1 type of macrophages. So those macrophages, they are inflammatory macrophages. And so we are characterizing more on these cells. And so this is the what actually I can share with you right now. So the next question we asked that whether these mice can show post-exceptional malaise, which is a very key pathological hallmark of ME/CFS. So to do that, we let these mouse run on treadmill. So this is before pre-treadmill activity of these mice for the vehicle and SIM-fed mice. And we can see that vehicle-fed mice, they are totally active and they don't have any kind of movement deficit. Whereas SIM-fed mice, they have a partial movement deficit compared to vehicle-fed mice. However, when we make them run in a treadmill for 14 RPM per 15 minutes, immediate after that, both vehicle and SIM-fed mouse, they have a transient fatigue that is quite expected. However, these mice, they have more fatigue immediate after the treadmill exercise. And then we gave them 2 days of rest. And after 2 days, we saw that these vehicle-fed mice, they completely regained their movement, whereas these sim-fed mice, they are still very much inactive, and they have still movement deficit. So this is kind of very intriguing and very interesting finding, actually, what we found so far. And we are doing more molecular study on that, on these mouse. So what kind of inflammation is happening, and if there any inflammatory demyelination in the muscle nerves. So all these analyses are going on right now. But what I can say that there are some limitations of this drug-induced model, because the fatigue is transient, because those mice, they come back to the normal schedule after like a 1 month. And the direct role of ATG13 is not clear in this mouse, because the drugs seem that can inhibit a different other targets, so that we don't know. So to nullify these drawbacks, we plan to do a ATG13 knockout mouse model, and where we are expecting the fatigue is much more stable, and where we can establish the direct role of ATG13. But on the other hand, that ATG13 whole body knockout mouse, they also show some kind of growth deficit, cardiomyopathy, and less viability. So these might confound the understanding of ATG13's role in ME/CFS. So that's why we just apply, and we got a recent R21 NIH grant from NINDS, where we want to make some kind of tissue-specific knockdown of ATG13 with this system, TLOX system. So we will do muscle-specific mutation of ATG13, brain-specific, and peripheral nervous system-specific knocking down of ATG13. So what we expect? We expect that ATG13 muscle knockout mouse, they will show some fatigue, maybe some post-exertional malaise. Whereas ATG13 brain knockout mouse, they might show some kind of anxiety and attention deficit, and ATG13 PNS knockout mouse, they might show some sort of dysautonomia. So that is our expectation, but time will say that what will happen in next few months and few years. And so thank you all for your attention. And this is our core team of our animal study. Our executive director, Dr. Gunnar Gottschalk, and this is Dr. Leigh Arnold from University of Wisconsin-Milwaukee. Linda is she's our attending veterinarian who is helping tirelessly in our animal studies. And our collaborators are University of Wisconsin-Milwaukee and COPA Laboratories. And our current study is being funded by SolveME and NINDS. So thank you all for your attention. Thank you.
Great. Thank you so much for that excellent presentation. We will now transition to the question and answer session and open the call for your questions. As a reminder, if you have a question for our speakers, please select the raise hand button at the bottom of your Zoom screen and we will call on you to unmute. If you are joining us by phone, please dial *9 to raise lower your hand and *6 to mute and unmute. You can also submit questions using the Q&A box. We will start by taking questions for Drs. Roy and Gottschalk. And I actually saw a couple questions for them in the Q&A box, so we can start there. Dr. Roy, can you say more about mitochondrial toxicity, potential causes and consequences, and how it might trigger ME/CFS?
Yeah, so mitochondria is our energy house, so that produce a lot of energy molecules known as ATP, or adenosine triphosphate. So when there is a mitochondrial toxicity, so the first thing what will happen, there is less production of energy molecules, ATP. So that can cause a very fundamental toxicity in a cell system. So mitochondria is kind of present in almost all type of cell types, in heart, in brain, and also in muscle cells. So when there is a less ATP production, when there is less mitochondrial function. So then we also expect, like when there is a mitochondrial dysfunction, there will be production of reactive oxygen species in the system. So that can trigger cytotoxicity, cell loss, inflammation in the system. So if you look at inflammation, that is directly involved to the mitochondrial toxicity. And the loss of energy also is involved in the mitochondrial toxicity. So these factors, they're very integral and in the survival and growth of any kind of, on the function of any kind of tissue. So that's why mitochondrial is very critical. Mitochondrial health is very critical in health and disease.
Great, thank you. Another question for you. Why not consider an inducible knockout model?
Why not consider, can you repeat the question again?
Why not consider an inducible knockout model?
Yeah, that's what actually we are planning to do. That like the Prelox system, that is the inducible knockout model. So where we will inject mouse with a drug named tamoxifen. So when we inject the drug, then the Prelox recombination happen, and the ATG13 will be knocked out specifically in an adult mouse, and it can be induced by the drug. So that particular model is very effective in order to nullify all the correlative deficits. So we are definitely, we have a plan to do that.
Okay, great, thank you. And can you talk about how the study in mice can possibly lead to treatment options for people with ME?
So once we establish the disease model, our next target will be to cure that disease with the application of different drugs. And those drugs, they can modify a different metabolic pathways. One of them definitely is the autophagy, so autophagy related proteins. mTOR is a target. mTOR is one of our target that we can definitely, we want to treat those disease mouse with some mTOR manipulator, mTOR modulator that can eventually ameliorate the disease progression, and that can improve the pathology, that can improve, that can protect the pathology. And the second option, actually, we also try to develop some kind of like a therapy, some type of like a peptides that also can like interfere in different inflammatory pathways in this disease model, so that it can improve the disease pathology. It can protect the disease pathology. So that's what our plan. Yes. So definitely the next step is to design some drugs to prevent this kind of like a disease progression in mouse. And after that, we will go to the preclinical stage, and definitely we have a long plan after that.
Great, thank you. I see several people have their hands raised. Scott Daniska, go ahead and unmute.
Hi, yes, can you hear me?
Yes.
Hi, so I wanted to ask, thank you for all your presentations, first of all, and Avik, your analysis on autophagy was really interesting. But one of the things that you mentioned is a question that you left is what is the trigger for these change, for these altered levels? And so I'm wondering, if there's been thought as to underlying drivers of CFS and what can be done to look for these underlying causes. And so there's been, a lot of talk about, hidden viruses, things like that. But a lot of patients, myself included, complain of things like toxic mold, VOCs in homes, renovations, etc., triggering a lot of their symptoms. And so I'm wondering if there's any thought or, prospective studies that you can think of to begin to analyze the environments that people with CFS are living in and be able to correlate, for example, household products with the emergence of CFS to really find an underlying driver? Thank you.
Yeah.
Thank you so much, Scott, for your question. As I mentioned before, that ME/CFS is highly heterogeneous disease. So it's like in terms of its etiology, it's very heterogeneous, and it's really hard to say that what causes this abnormality. However, we have to start from something, from some common reference point. And as a molecular biologist, we can target only a pathway in a cell, and we can start from that reference point, that like if that pathway is modulated or if that pathway is manipulated, so what are the consequences that can happen? So, and from our preliminary study, from our pilot study, we saw that ATG13 is a protein which is severely metabolism of that protein is severely impaired. So that gives us an indication that the autophagy impairment is very critical in the progression of this disease. And yes, autophagy is a pathway that is linked to all different sorts of metabolic pathways like inflammation, mitochondrial toxicity, apoptosis, all different things, all different metabolic processes. So right now in our understanding, that mTOR pathway or the autophagy, mTOR mediated autophagy pathway is a very common reference point from where we can start our disease model study, and where we can manipulate that particular pathway to understand, to identify a drug candidate. And really, it's very hard to say that what exactly is causing the autophagy impairment, and what. Yes, you are right, because you said that that mold attack, mold or viral infection. Yes, they might be involved, but as I said, it's highly heterogeneous in nature, and it's very difficult to say what exactly causes that autophagy impairment. So that's my understanding.
Great, thank you. Deborah Jerica, please unmute and ask your question.
Can you hear me? Yeah, hi there. Dr. Roy, you already answered my question. I asked why not use an inducible model before you mentioned Crelox-P, so that's it. Thank you.
Okay, thank you. Okay, Court Johnson, you want to unmute and ask your question, please?
Yes, thank you. Hi. Hi, Avik.
Hi, Court. How are you?
I'm good. I'm good. So with regards to, I thought the macrophage infiltration into the muscles was so interesting. Could you do a muscle biopsy or something like that in ME/CFS and see if the same thing is?
I'm glad to do that. Yes.
Oh, good. And with regard to mTOR inactivating drugs, what drugs would there are mTOR inactivating drugs available now?
Yes. So rapamycin is one of them.
Okay, good.
But again, it depends on what stage of the disease. And in my case, when I did my pilot mouse study, we saw that young female. So we started with two different age groups. One is 3 to 4 weeks old mouse groups, and another is 5 to 6 weeks old mouse group. And what I found that 3 to 4 weeks old animals, only female mice, they are susceptible to the mTOR activation mediated muscle fatigue. Whereas 5 to 6 weeks old female mouse, they these mice, they didn't show any kind of pathology after mTOR activation. So it's young female. So those are more susceptible for this kind of model, female mice.
Got it. Interesting.
if I can jump in, studying muscle biopsies from individuals with ME/CFS is part of the new funded Cornell Center. So you'll probably be seeing they'll be recruiting individuals for those studies soon. So I know that that's part of their upcoming plans.
Can we have some muscle tissue from them, like if we want to?
I'll put you in touch with them. Yep.
Yeah. I think we'll figure it out. Yeah, I know.
Great, thank you. There's another question in the QA box. Will you also be looking for changes in sensory hypersensitivity?
Avik, I can maybe take that. I think one of the beauties of the CRELOX-P model with the tissue-specific knockout of ATG13, for example, or any other target, is that you can target specifically like neurons only, or peripheral nervous tissue only, or muscle tissue only. Those phenotypes that are described there, hypersensitivity, et cetera, you know, in neuroscience and other places, they're pretty well described in animals to some extent. But again, we are dealing with mice, not humans. So we're going to do our best to see if we can develop and find some phenotypes that may be correlarily marked towards what we see in human patients as well.
Great, thank you. Okay, Ruhi Snyder, please unmute and ask your question.
Hi, can you hear me?
Yes.
Yes.
Thank you so much. I really appreciate it. I just want to focus on demyelination a little bit. You're talking about demyelination of the muscles, as well as, I think from my research that I'm gathering, is demyelination within the brain itself, so the white matter. My deep concern is the fact that the young humans who are suffering now from ME/CFS, this is an incredibly important time in terms of their brain development, especially in terms of myelination. And is there anything that we can do at all at the moment to stop that kind of process that is being generated because of demyelination that's happening both in the muscles as well as in the brain?
Yes. So if you look at the CNS demyelinating disease, like multiple sclerosis and ALS, amyotrophic lateral sclerosis. So all these diseases, they have a multiple therapies, and so those are available. So you can definitely consult with any specialist with multiple sclerosis. Like if you are interested about demyelination, so any clinician, any physician of multiple sclerosis who are expert in multiple sclerosis and ALS. And in our previous research, we have shown that cinnamon, which is a herbal spice, we usually use in our curries and all these dishes. So cinnamon is very important herbs that prevents demyelination, that protects myelin loss, and at least in our mouse model studies. And we have initiated a clinical trial in Ross University Medical Center in Chicago, where actually a large number of patients, MS and ALS patients, were recruited in that study. And the study is going on right now, the cinnamon treatment. And so that's what I can say. Yes.
Great, thank you. We do see somebody who is calling in on the phone with their hand raised, the phone number ending 125. You can go ahead and unmute and ask your question, please.
Yes, hi. Hello.
Hi.
My name is Eileen Holderman. I'm an advocate and former member of this advisory committee to HHS in Washington. I'd like to thank everyone for the call and their presentations. My comment and question is for Dr. Nath. At the beginning of your NIH Intramural study, the criteria for recruitment changed 6 times, and we brought that to NIH's attention. So my question is, did you use the expert's definition or criteria, the MEICC? If not, why not? And can you comment on the extreme concern advocates are having over the criteria issue, such as the creation of SEID by the IOM and the recent coding of that on par with ME, and the trend towards studying and focusing on one symptom of this vast disease, which is PEM? Thank you.
Yeah, well, thanks for bringing that issue up. Yes, so we put together a panel of experts that adjudicated every case that we enrolled. So we made sure that we had several different criteria and that these patients met each one of them. So we are very confident that these patients really had ME/CFS, and it was a post-infectious ME/CFS. So we screened hundreds of patients to enroll the right patient population. So, so I'm pretty confident we have the right patient population.
Thank you. I have another question in the QA box. This one will be for Dr. Whittemore. It's about the research roadmap. In the past, patient representative feedback to HHS has not been substantially incorporated. How do you plan to ensure that patient input from this working group will have tangible impact on the direction of your work and not be solely symbolic?
Yeah, thank you for that question. So it's really the reason why we have involved so many individuals with lived experience, both on the working group of council, as well as in helping us to plan the webinars and to be actively participate in the discussion and formation of the research priorities. So in a previous experience I had working at a nonprofit where the hallmark symptom of the children with that disease are seizures, we were heading down a path of planning a lot of research on epilepsy and seizures in that genetic disorder when the input we got from parents and individuals with the disease was the cognitive behavioral issues that were more important to them. And it really shifted the way we thought about moving things forward. So it's really that kind of critical input in hearing from the community about what is most important and critical to you that will help to shape things going forward. So it is not at all going to be symbolic, and we are here and will provide many different opportunities for input and feedback from the community. So thank you.
Great, thank you. We are almost at time, but we have time for one more question. Yuval Sheffi, you want to unmute and ask your question, please?
Hey, do you hear me?
Yes.
Okay, so I wanted to ask if the weakness of the muscles that we're also dealing with is part of the PEM search, or it's another thing to investigate.
Dr. Nath, if you want to take that one.
I'm not sure I really understood the question. So it's not really weakness per se in the muscle. It's more of exhaustion after exercise intolerance that these patients deal with. Weakness is a very different aspect. That means there's either muscle pathology or the nerves themselves are damaged. So, for example, you have a stroke or something, you could get weakness. So I don't think that ME/CFS is really a disease of muscle weakness.
Dr. Nath, do you see muscle weakness though after deconditioning? So say for someone that's been
that's very different.
Bound for
that's not what, at least in neurological terms, I would call weakness.
Right.
That is just fatigue and exhaustion and exercise intolerance. So, which is very different.
Great, thank you. It looks like we're right at time. Thank you again, Dr. Roy and Dr. Gottschalk, for joining us to talk about your research today. A recording and transcript of the webinar will be posted to the NIH ME/CFS website soon. In closing today's telebriefing, I'd like to remind you about our listserv for updates from NIH. To be added to the listserv, please visit the NIH MECFS website, which is www.nih.gov/MECFS, and click on Join Our Listserv. Thanks everyone for joining us today and for the thoughtful discussions. Have a great afternoon. Thank you.